So this is a collaboration I'm working on with folks up at the Allen Institute for Cell Science. And I'm not a biologist myself, but all of my friends who are tell me that the Allen Institute is producing absolutely the best image data set in the world. And so what they're doing is they're taking these in-cell pictures, trying to keep the cell as much alive and as healthy as possible, and to take really detailed microscopy images of all of the internal structures of the cell. And what we're helping them do is try to make this data available to scientists all over the world for collaboration in a reproducible and versioned way, so that when they publish papers, they can reference a specific set of cells. What the features that they need for this effort is specifically they needed really scalable data versioning, and they need high bandwidth access because the data sets are at least, maybe not large by this group's standards, but they're still pretty big data sets. And the final requirement is that most of these scientists that are working with this, we think of images as pictures to consume with our eyes. But in fact, most of the work that's being done with these images is actually being done looking at them as NumPy arrays and analyzing them in Python in toolkits like scikit-learn and TensorFlow and PyTorch working in Jupyter notebooks. So what they've done is they've come and decided to use a system of version data packages. These are bundles of data and metadata that can be transmitted together. It gives them deduplication to give them more efficiency in their space and bandwidth. And most importantly, it's a lightweight metadata-only service that gets out of the way when it comes to transmitting the actual large data blob. So there's a direct connection to the high-performance, high-bandwidth cloud storage, and it's open source. In essence, you can think of it as working, this open source library working like Docker. The workflow is essentially build a data package, create this metadata wrapper around your raw data, push that, you can push it up to a hub like Docker Hub, install and import, which is exactly what they do. So in their work, there's scientists doing their wet work are taking images, new images every day. Then the researchers in their informatics department can build a view, build a data set with a particular use case in mind, with a particular study, or for a particular paper. Creating a new view of these data sets is, again, a metadata-only operation, so it can be very fast. And they can push them up when they're ready for publication. The package itself borrows from techniques and other data versioning schemes. It's essentially a hash tree. The manifest contains the contents and all of the individual SHA-256 hashes of the raw data fragments. The data fragments themselves are then stored by their hash so they can be deduplicated and transmitted independently. This is what it looks like when the Allen Institute is publishing one of their packages. There's a nice web catalog. You can go online and browse their data. Then when consumers want to interact with this and when they want to, as I said, the key is to getting the data quickly is to just essentially get out of the way. The registry handles permissions and handles versioning, handing out essentially the right manifest, which is again the right uniquely identified fragments. And then from there, it's straight to S3. The last thing the package does is it's this metadata wrapper which lets you interact with a large data set right from your laptop, right from Jupyter. So by importing just the metadata, you can go right in IPython and data scientists can even be tab completing their way through a data set that would have no chance of fitting in their environment. Then when they want to actually interact with the images, they can download just the fragments that meet their needs. And if for more information, everything is open source and on the web. Please check out our website at quiltdata.com or our GitHub repository, quiltdata.quilt. And please send links to all of your cellular biology friends for this really amazing image set at allencell.org.